All right, so my presentation is titled Development of Novel ROS1 Inhibitors to Overcome Resistance of the Current Drug, lorlatinib. So a little background, ROS1 is the c-ros oncogene 1. The PDB ID is 4UXL, which is pictured here. It's expressed in a wide range of cancers, but our particular focus here is non-small cell lung cancer. And this protein/gene was the target of this first generation inhibitor right here, developed by Pfizer in 2011. That's crizotinib. So that mainly applied to the wild type, but as we know, genes mutate. So that worked for a little while, but researchers found that patients were developing resistance to that drug. So another team from Pfizer in 2015 developed this drug here, lorlatinib, which applied mostly to these mutations. So a little background on the research methods that we used in the dry lab. We did the database search mainly with Protein Data Bank and Protein Data Bank in Europe. We looked at the structural properties. We did homology modeling, which is basically making a 3D structure of it. We docked the ligand to the protein, and we also ran virtual screening using a list of FDA-approved drugs. So I won't go into detail on all these due to time constraints, but I think the most important was docking or the virtual screening. So here's the top 5 we got, and they are in descending order of how good their docking score is. Basically, negative the better, more negative the better. And I apologize for the error here. This should be lorlatinib, not wild type, but the important thing to note is that all of these have a better docking score than lorlatinib there. So applying these methods to the new drugs, we ran the EGFR osimertinib assay. And basically what we did was we mixed the protein with ATP and the inhibitor. And what happened there was a luciferase reaction. This is just the general reaction scheme I found. The most important thing is ATP is reacting here, and one of our products is light. So we ran that assay, then we measured luminescence to determine the level of inhibition. More luminescence, more inhibition. And our aim would be for the new drugs would be to get a lower IC50 than lorlatinib, which is 1.3 nanomolar. So we take that information, we'd apply it to we swap in basically ROS1 and then our top 5 compounds, and we would expect to see more inhibition due to the better docking scores compared to lorlatinib. So I got.
So this is similar to the one we did in wet lab, right?
Yeah.
Can you explain like the difference between this and that one?
I'll be honest, that's something I have to research more. I didn't look too much into that. But basically I figured the procedure would be about the same because it's just swapping in different proteins and inhibitors.
Okay, you go to your this slide, go to the okay, here. I just one comment. I like this data because this data you didn't show the supporting why that mutation causes drug resistance. You have the study actually provide evidence supporting those 2 mutations cause drug resistance. And so, yeah, as a comment, I like this because everything in the science you want to back up by some facts based on some data, not based on opinion, right? So this is a good example that you show that you found the literature data supporting this mutation cause problem. Then our next goal is to find compound targeting those mutations, right? So yeah, that's common one. Again, it's A. Okay, so next.